Now, I'd like to introduce our next speaker, Dr. Lakshmi Narayan Mishra. Dr. Lakshmi Narayan Mishra is currently a postdoctoral scientist at the Albert Einstein College of Medicine, New York, USA. He was awarded PhD in chromatin biology under the guidance of Professor M.R.S. Rao, former President, Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore. His work focuses on understanding the role of linker histones in regulating the gene expression. He has published several research articles in international peer-reviewed journals of high repute, including in the Proceedings of the National Academy of Science, USA, Journal of Biological Chemistry, USA, Nature Scientific Reports, Epigenetics and Chromatin, Biochemical Journal, Journal of Proteomics, etc. For his extraordinary contribution in the field of epigenetics and chromatin, he has been awarded Harry Eagle Scholarship for the Outstanding Postdoctoral Fellows by Albert Einstein College of Medicine, New York, USA. He is a recipient of several international scholarships and travel grants. He is an early career reviewer, board member of the Journal of Biological Chemistry, USA, and is also serving as an associate editor of the Bioprotocols, an international peer-reviewed journal published by the Research of Stanford University, USA. Now, I'd like to invite you, sir, to please enlighten us with your thoughts.
Okay, so thank you for the nice introduction and thank you for inviting me to be one speaker in this international conference. So today I'm going to talk about linker histones, and I would try to cover it from my PhD to my postdoctoral journey that I am doing still now. So this is the overview of my presentation. So first I will introduce all of you to the chromatin, and then I will discuss about the functions of linker histone in spermiogenesis. That is one process when by which sperms are formed. And then I will go on discussing about the role of linker histones in controlling the chromatin accessibility. Role of linker histones in modulating core histone PTMs, and then I will summarize, and then we will have some question answer session. So what is DNA? How DNA is packed inside the nucleus? So all of us know that DNA is the genetic material, meaning it carries all the information that is needed to form a body, all the system processes that happens in the body, everything is there encoded inside the DNA. And it has only 4 bases, A, T, G, and C. So adenine, thymine, guanine, and cytosine. If you see the total length of DNA in each cell, it is almost like 2 meters long. However, it has to fit inside the nucleus that has the diameter of only 10 microns. So how is it possible that you can fit 2 meter long DNA in each cell in 10 micron diameter nucleus? So it's not feasible in general case. So there are several types of proteins called histone proteins and non-histone proteins that help in this fitting process. So these histone proteins are of 5 types: H2A, H2B, H3, and H4. We also call them as core histones. And there are linker histones. They are 11 subtypes in mammals, and they are called linker histone H1s. So this gene expression depends on how chromatin structure changes with time or when they get any stimulus. So this chromatin accessibility plays a major role in gene expression. So what is gene expression? I will come later on. So if we see on the right side the structure of DNA, so this is the DNA, and when it has to fit inside the nucleus, it cannot fit alone. So it has to bind to the histone proteins. You can see here on number 3. So this association of histone protein with DNA the structure which is formed by their association is called nucleosome, and nucleosome are also called the structural and functional unit of chromatin. So DNA inside the nucleus is present in the form of chromatin, and the structural unit of chromatin is called nucleosome. So we have to remember this throughout my presentation. So there is a fifth kind of histone that I mentioned, H1. This H1 is found outside the nucleosome, and it is bound to the linker DNA. So what is linker DNA? Linker DNA binds two nucleosomes together. So linker DNA binds two nucleosomes together, and linker histone, the name as it suggests, it is found in the linker DNA, and it helps in the folding of these nucleosomes on each other. So H1 is essential for chromatin condensation. It condenses. It is no longer like it's not longer open. It is getting closed when H1 is bound to the nucleosomes, and these structures further condense with the help of nucleosome histone tail interactions. So all these H2A, H2B, H3, and H4 have N-terminal tails that is protruding outside the nucleosome, and they interact with each other and help in the formation of condensed chromatin, and H1 adds to that condensation. And this is the mechanism by which all the DNA packs inside a tiny nucleus. So how DNA passes from parents to offspring? As I mentioned, that all the information is inside the DNA. So this has to pass from generation to generation. Whatever I have got the information that has come from either from my mother or my father. So what carries all the information? And the answer is DNA. So DNA is passed through the through the sperm that goes in and mixes in the form of pronuclei, sperm pronuclei with the egg pronuclei, and fertilization, it forms a zygote, and zygote has all the information. It's a totipotent cell that can form all the body part of the mammalian system or any living system that has sexual reproduction. And then offspring are formed, and these offsprings had DNA that has come from either the father or the mother. So this DNA has the capacity to form all the body parts. So how is it possible? There were only two cells. Sperm is haploid cell, egg is haploid cell. When they mix together, means fertilization happens, it forms a diploid cell, that is zygote. And then zygote has all the memory. So what is there to give the memory? So there are, for the memory, there are histones that has been retained during this process, a spermatogenesis process that I will discuss. That is my research actually. And ovum also gives some information about what to form, which body part to form. So everything is there. All the information is there in the zygote. So then comes the flow of information, how the organs will be formed. So this is called central dogma in molecular biology. Means DNA forms RNA by the process called transcription, and RNA forms protein by the process of translation. And this process is called gene expression. Means DNA is giving a functional product either in the form of RNA or in the form of protein. And since DNA, as I mentioned, is bound to histone proteins or non-histone proteins or even RNA, they are inaccessible to any other transcription factors or any other transacting factors. No transacting factor can bind to DNA if it is bound to histone protein or non-histone protein that has much more affinity. So no one wants to leave their friend, and DNA and histones are friends. So these DNA are not accessible. So to make them accessible, histone has to go away. Otherwise, there won't be any friendship with other transacting factors. So chromatin accessibility plays a major role in transcription process. So how will you change the chromatin accessibility? So there are three different mechanisms by which you can change the chromatin structure. First is chromatin composition. So as I mentioned, that nucleosome is the structural and functional unit of chromatin. So you change the nucleosome. So how will you change the nucleosome? So as I mentioned, that there are five histones 5 types of histone: H2A, H2B, H3, and H4, and H1. So you change those histones, canonical histones, with variant histones. There are several variant histones for H1. I mentioned 11 histone variants are there for linker. There are no linker and there are no variants for histone H4, but H2A has many histone variants. So all the histone variant have different characteristics. Although they look similar, there are 3 or 4 amino acid changes, but their structure and function are completely different. So 1 or 2 amino acids can change everything inside the nucleus. That's by mutation or substitution, anything can happen and function is going to change. So we change the composition or we modify them. So histones can be modified by post-translational modifications, and those post-translational modifications are covalent modifications such as acetylation, methylation, phosphorylation, sumoylation. There are umpteen number of modifications that can happen, and they can change the structure and function of the histone, thereby changing the accessibility also of the chromatin structure. And third is by changing the position of the regulatory elements. So for example, regulatory element is here and it is bound to the histone. No other protein can come and bind to these regulatory elements. So there are modifiers called ATP dependent chromatin remodeling factors which can remodel them. So how they will remodel? They will remodel in such a way that these nucleosomes can slide little bit. They start moving with the help of ATP, because ATP is the energy source there, and these will be, after some time, these blue region inside the nucleosome will be accessible and regulatory elements will come and bind, regulatory factors can come and bind here. So these are 3 mechanisms which I will be talking about today. So as I mentioned, there are several H1 variants or linker histone variants. These are of 2 types, somatic and tissue specific. And tissue specific histone variant was my project during PhD at JNCSR Bangalore. These tissue specific histone variants are of again 2 types, because there are 2 types of tissue based on the mammalian sex. So these are testis specific or oocyte specific. So oocyte specific are only found in female. Testis specific are found only in males in the testis. So testis specific are again of 3 types: H1T, H1T2, and HILS1. HILS1 was the newest member when I joined my PhD program in 2006, and this protein was reported only in 2003 with no known functions of this protein. So these were the properties that were known at that time when I joined the PhD program. Only the first 3 facts about this protein. So this is a 19.2 kilodalton protein, expresses in elongating and elongated spermatids. So these are the steps, and these are the shapes of the spermatid during maturation into mature form. Interacts with transition protein and protamine. I will discuss about this in next slide. And then someone in 2007 lab showed that this protein has significantly lower level of HILS1, has lower level of transcript, and that was reported in infertile male that had asthenozoospermic condition, means motility was very less in those males, sperm motility, and sperm motility plays a major role in fertility. And transition protein 1 and HILS1 double knockout mice were infertile, means there is some property in this protein that plays a role in fertility. And when you go to a doctor and doctor says that, okay, yeah, everything is fine, there is no problem with the male or the female, but the female is not getting pregnant. What is the rule? What is the reason behind that? So nobody knows that what are different other proteins which are playing a role in that spermiogenesis process, and that was the purpose of studying this protein. As a biotechnologist, so when I joined biotechnology program, I was told that biotechnology is application of technology for the betterment of human life. So whenever we study any protein, any RNA, any DNA, we look for some application. So I found that there is application of this protein in spermiogenesis or fertility. So HLS1, as I mentioned, it expresses during elongating and elongated spermatid, and that is also the step. These are the elongating and elongated spermatids, and these are also the steps when chromatin remodeling takes place. So what is chromatin remodeling? So chromatin remodeling is when most of the histones that we know, that I mentioned also, that histones are bound to DNA, but there are exceptions. A sperm is one exception where most of them, 95% of the histones are replaced with first with transition proteins. These are called transition protein 1 and 2, and they are further replaced with more basic protein called protamines. So what is the necessity of these histones are there in all of the body cells? Why a sperm has different protein? Because this sperm has to travel inside the female reproductive tract, and the female reproductive tract has a pH of 3.5 to 4. It is acidic. A sperm can get damaged if it is not condensed, highly condensed. So it has to condense to spare itself from DNA damaging agents. So if you read about the sperm or ejaculated sperm, you will see that the pH is alkaline, slightly alkaline, and that is because a sperm within 30 minutes it will be neutralized, and the female cannot get pregnant because at least one sperm has to go and meet with the egg. And there are like 50 million or 60 million sperm, but to the surprise, only one sperm meets with the ovum. So, and if it does not, even one cannot, it is called infertility. So since this protein plays a role in infertility, it is worthwhile we should know about infertility. What is infertility? So infertility is inability to achieve pregnancy even after 1 year of having regular unprotected intercourse, or after 6 months if the woman is older than 35 years of age. Most, like out of 10, 1 or 2 people are suffering for infertility. Nobody tells, but it's the reality. It affects an estimated 15% of couples globally. It's everywhere, in US, in India, in Japan, in China, everywhere. Many different medical conditions and other factors can contribute to fertility problems, and every individual may have single cause or multiple cause on infertility, and there may be some unidentifiable cause also. For example, if the protein level is low, doctor cannot tell that this protein level is low because it's very costly business for them also, and there are no like studies showing that okay, you can study the expression of this protein. You will have to take a sperm in different spermatocytes in different phases of maturation, and nobody wants to give the sperm in different phases because you have to cut the testes. It's very difficult to do that. In 1/3 of the infertile couples, the problem is with man. 1/3 of the infertile couples, the problem is with the woman. And in 1/3 of the infertile couples, the problem cannot be identified or is with both the man and the woman. So in our society, we used to know that, okay, female is the culprit. She cannot get pregnant. But remember, it's equal chance of infertility in man and woman. So possible cause of female infertility is failure to ovulate, accounts for almost 40% of female infertility. This is the most prominent cause, that female is not ovulating. Number of ovums are limited in females. Problems in the menstrual cycle, structural problems of the reproductive system such as fallopian tube and uterus problem, infections such as untreated gonorrhea and chlamydia, implantation failure means embryo is there but it's not getting implanted inside the uterus. Endometriosis means this is endometrium is the lining of the uterus that gets shredded with time if there is some problem. Polycystic ovarian syndrome, if I know correctly, it's like out of 10, 3 women facing this problem, polycystic ovarian syndrome, primary ovary insufficiency, uterine fibroids, and autoimmune disorders. If you come to the male infertility side, there are abnormal sperm production or function due to the undescended testicles. Testicles has to descend from the body because it needs 2 degrees Celsius less temperature from the compared to the body. We have 37, and a sperm matures at 35 or 36 maximum. It cannot mature at 37 degrees Celsius. Genetic defects, health problems such as diabetes or infections such as chlamydia, gonorrhea, mumps, or HIV. So please, everybody, take care of your health. Don't be diabetic. It causes infertility in future. Enlarged veins in the testes, blood supply changes completely, problems with the delivery of a sperm such as premature ejaculation. This is most prominent cause of infertility. Certain genetic diseases, structural problems such as blockage in the testicles. You have a sperm, but it is not coming in the serum, so that's a problem. Or damage or injury to the reproductive organs, pesticides, radiation, cigarette smoking, alcohol, marijuana, antihypertensives, and anabolic steroids that people take very usually. They are all causes of male infertility. Damage related to cancer and its treatment, including radiation and chemotherapy, they all cause infertility. So infertility, as I told, is everywhere, and these are the males contributing to infertility worldwide. In Asia, we have 30% males, 37% of all the infertile couples, 37% are males. In US, you can see that in North America, it's 50%, it's much more. In Middle East, it's even 60 to 70%. So it keeps on changing. So finally, so fertilization. So if the male is fertile, woman is fertile, so what will happen is fertilization is the process where ovum, that is female gamete, and male gamete is a sperm, they meet together and they form zygote, and only one sperm goes inside the ovum, and this is a sperm ovum nucleus, and this is the nucleus of the sperm, and only the nucleus part goes from the sperm, not all the parts. So this nucleus is highly condensed in this, and they have 95% transition protamines, and only 5% are histones that we call residual histones. So my protein comes into play now. So HLS1, as I mentioned, that it was reported in elongating and elongated spermatids. So the first thing, because the properties were not known, so we have to correlate with some property. Someone takes birth in our family. What we do is, my mother comes or my father comes, and they tell that, okay, this looks like this looks like mother, or this looks like father. So in biology also, when we do research, we go towards the family. So family business is the main thing everywhere. You have to correlate. So HLS1, I wanted to see how much similar this is with other linker histones which has been reported in the literature. So all the linker histones have tripartite structure, means 3 parts are there. N-terminal histones that has no structure, globular domain that has a structure, and C-terminal domain that does not have a structure. And most of the functions are done by C-terminal domain of the linker histones. They condense the chromatin, and remember, the main property of a linker histone is to condense the chromatin. So what I did is, I first assembled the sequence of rat H1D. So human is not possible to study for all these types of work. So you have to take some animal, right? So either mouse or human, rat, those are very similar. They are physiologically similar to human. So I took rat. The reason was we get much more like bigger testes compared to mouse. Mouse is very small animal compared to rat. So rat I took for my study, and my lab in IISc Bangalore, Professor M.R.S. Rao's lab, that has worked extensively with rat H1D and H1T. So we had experience of working with these proteins, except with HLS1 and H1T2. So first thing I did was I assembled their sequence and found that, oh, there are differences in many of the regions. This is N-terminal domain, red region is the globular domain, and again there is C-terminal domain, and in the C-terminal domain, SP or TPKK motif plays a role in chromatin condensation. So TPKK, if you see this TPKK, all the TPKK or SPKK motifs are completely different in HIRS1. If you see the sequence, this is completely different. So one thing I knew, that, okay, based on the literature search and my background analysis, it cannot be a strong condenser of chromatin, because chromatin condensation motifs are completely different in this protein. And second is DNA binding protein. If it does not bind to DNA, how can it condense the chromatin? So for DNA binding, globular domain is responsible. And in the globular domain also, we could see that 3 amino acids have changed out of 7 amino acids. So it will have altered DNA binding capacity. So the main thing that people want to know about the linker histone property is chromatin condensation property. And as I mentioned, the C-terminal domain was completely different. So I studied DNA binding property, I studied DNA condensation property, I studied chromatin condensation property, and I'm showing here the chromatin condensation part. So what I did is I took the testes, I isolated native chromatin and I stripped all H1s from there. So there is no H1 now. And then I added back H1D, H1T, and HLS1. So why did I take H1D and H1T? These are reference for me, because these are working as a reference or control for me, because H1D is a strong condenser of chromatin, H1T is a poor condenser of chromatin, and HLS1 property we don't know. So either it will be a poor condenser of chromatin, its property will be similar to H1T. If it is a strong condenser of chromatin, its property will be similar to H1D. So these are serving as a control here. And then I analyze chromatin condensation by circular dichroism spectroscopy. And here you see it's a simple view of the result that I got. So if these are molar ellipticity on y-axis, and x-axis are different chromatins. So native chromatin is much more condensed, and to understand this figure, you have to remember that the molar ellipticity is less, means chromatin is more condensed. If the molar ellipticity is more, it is less condensed. So as you see, native chromatin, where H1 is there, all the H1s are there in the testis chromatin that is native, it is more condensed because the ellipticity, molar ellipticity is less. As soon as I strip H1, H1 is not there, means there is no protein to condense them, the ellipticity goes high, means it is less condensed. When I add one H1D per nucleosome, molar, this is molar ratio, it comes back, means it condenses again. So H1D is a strong condenser of chromatin because it is similar to native chromatin, a molar ellipticity. But when I took out H1T, when I took out H1T, when I took out H1, molar ellipticity increased, means it was poor, it was not condensed. But when I added H1T, it was further like less condensed, it was like more open. And similar was the case with HLS1. So we found that HLS1, hello, is my time over? Hello? Hello? Okay. So HLS1 is a poor condenser of chromatin. So next thing was, okay, so as I mentioned that histone post-translational modifications also play a role in these processes. So I wanted to know how, like, what are the modifications in this protein. So I separated the testis histones by RP-HPLC, and this is the here is the peak for HLS1. And this study we published in Journal of Proteomics, and we found that there are several residues which undergoes post-translational modifications. These modifications are known in other proteins, not in this, but these were known. So another modification that was serendipity for me, I was just testing several antibodies, and I found that tyrosine and phosphotyrosine antibody also reacted with HLS1 protein and other linker histones also. And this was surprising that how tyrosine phosphorylation is happening in linker histones when there is no report like this. So this was the first report that linker histones undergoes tyrosine phosphorylation, and as a control for that, I had used phosphorylated tyrosine BSA, and as a negative control, I used BSA that has no phosphorylation, and this confirmed this. This was further confirmed by tyrosine a specific phosphotyrosine a specific HLS1 antibody. So then the question is, where does it bind in the genome? You have to know the location of this protein. So HLS1, we did chip sequencing, And chip sequencing showed that it binds primarily with the intergenic and intronic regions of the genome. So why it binds to intergenic and intronic regions? So I looked in the literature and I found that most of the histones, that 5% residual histones, are there where intergenic and intronic regions are present. So those 5% regions which has residual proteins, histone proteins, and that will give signal for the body development when fertilization happens and like child gets birth, takes birth. So throughout the life, starting from the fertilization up to 9 months in the as a fetus, and then throughout the life, there are histones which keeps on giving the signal that okay, this you have to do. This information has come from your mother or your father. So these are the regions which we found that okay, these associates with this protein. So summary of this part is HLS1 is a poor condenser of chromatin. It gets post-translationally modified by phosphorylation, acetylation, and methylation, and it associates primarily with repetitive DNA sequences. These are DNase1 hypersensitive sites and retain parental histones. So coming to the second part of my talk, that is a study of the effect of linker histones on chromatin accessibility. So nobody knew that how much chromatin condensation it can provide, linker histones can provide. So can we reconstitute nucleosomes in vitro? Nucleosomes are present in the body, but can we do outside the body? And the answer is yes. So I joined Professor Jeffrey Hess lab at University of Rochester, who is a world leader in reconstituting this system outside the body. And when we bind nucleosome positioning sequences, these are DNA with histone octamer, so two molecules of each of histone H2A, H2B, H3, and H4, it forms a bits on a string kind of a structure that we all must have studied. You also must have studied in any molecular biology book. So this is a bits on a string kind of a structure, and when we add 0.5 millimolar magnesium, it starts folding, and when we add H1 linker histone to that, it further folds, and at 10 millimolar magnesium, it is highly condensed in nature. So these are the this is the process which I described above. So how can we study chromatin accessibility? There are no method. So what I did after discussion with Jeff is I ligated 12 mer nucleosome array. So there are 12 nucleosomes in one system, and there is a central nucleosome which has DRA3 site on both the sides. And then I ligated these 12 mer nucleosome arrays from both the sides that also has DRA3 enzyme, restriction endonuclease site. So when we ligate together, it forms a continuous 25 mer array. And if I bind linker histone here and digest with this similar enzyme, DRA3 enzyme, if it is highly condensed, DRA3 cannot bind there. And the digestion rate will be very slow. If it is open, the digestion rate will be very high. So I did that, and as a control, everywhere in the biological system, you have to have one control. So as a control, I took control DNA, that is naked DNA, nothing. So because this was the linker DNA here, linker DNA we are digesting, so we have to take some DNA that is free of histones. So I took control DNA, and then this strategy we published in Nature Scientific Reports in 2016. So this is the strategy on the right side. What we did with this to study the chromatin accessibility, and we found that in 10 millimolar magnesium, when the chromatin is highly condensed, you can see here this structure, tertiary structure here, fully condensed, and in 0.5 millimolar magnesium, structure is like this, zigzag. So when we digested that, relative rate was almost like 280 times less when wild type chromatin was having H1 bound. So chromatin was not accessible at all. It was 280 times less. And in 0.5 millimolar magnesium, when the nucleosome had folded, it was still 50 times, almost 50 times less accessible. It means linker histone drastically decreases the linker DNA accessibility in partially or fully condensed array. And when there has to be gene expression, linker histone has to come out, otherwise there cannot be gene expression. So this we published in Journal of Biological Chemistry in 2018. So the last part is 2 slides more. what linker histone does more inside the body. So linker histones stimulate the activity of SUP39 enzyme and ZDB1 methyl transferases. So we reconstituted, when I joined the lab, I reconstituted dinucleosome, not 12, not 1, but this time I did dinucleosome, and I added or did not add the H1 to them. So when we added, you can see the red region here, this is H1 added. And then I did methyl transferase assay, means I added SUP39H1 enzyme, ZDB1 enzyme, and G9A enzyme, and allowed it to bind, methylate the histone H3, and they are known to bind and methylate H3K9, and they produce H3K9 dimethyl or trimethylation. And G9A is found in the euchromatic region. So there are two types of chromatin: euchromatin, that is getting expressed; heterochromatin, that is not getting expressed, means transcription won't be there. Euchromatin, transcription will be there. So G9A is found in the euchromatic region, and its activity was not affected. You can see that it's like the intensity of the band is same. But if you compare here, intensity of the band of H3K9 dimethylation or trimethylation was much more compared to when there was no H1. If you compare first lane with second lane, it means linker histone stimulates the activity of SUP39 and ZDB1. So this we published in Proceedings of the National Academy of Sciences this year. And then we also created embryonic stem cells that has very low level of H1 expressing. So this is very low level of H1 expressing, and these are measured satellites. And measured satellites should not express, but it was expressing because there was no H1. So expression was really like happening there. But when we added full length H1, black region, you can see the expression decreased immediately. Means H1 did not allow them to express, and that was the reason that I was studying all these things. And when we added H1D del 75, means del 75 means from the C-terminal domain, we have deleted 75 amino acids of the C-terminal domain. And if you remember, it's very difficult to remember, although it's new topic for many of you. So C-terminal domain was responsible for chromatin condensation. So there is no chromatin condensation, and so the expression will be more. So expression, it was here, expression was very less, it's much more now. It's more than 5 times. It has decreased 10 times, and now it has increased 5 times again. So this we published in PNAS this year. So the last slide is here. So this in this study we wanted to see what are other methyl transferases that gets stimulated or inhibited by linker histones. So this time we wanted to see the PRC2 activity, and if you see H1D, and this is delta CTD, means there is no CTD, we have clipped it off, and this is checking the activity of PRC2. As a control, loading control, we have H4 protein antibody. So Western blot was done with either H3K27 trimethylation antibody or H4 antibody. H3K27 trimethyl, because this protein PRC2 methylates H3K histone H3 at 27th residue, that is lysine, and it gives 3 methyl groups there. So without H1, you can see the band is faint. As soon as I add H1D, band is very intense, means it is stimulating the activity of the PRC2. But when we have del CTD, means CTD is not there, you can see that it's going faint, but not as faint as minus H1. So del CTD is responsible, but not it is not solely responsible for this active stimulation of PRC2 activity. And you can see the graph here again. So summary of my talk today is linker histones regulate the gene expression by restricting the accessibility of chromatin. Linker histones control the gene expression by chromatin condensation. Linker histones C-terminal domain play an important role in chromatin condensation. And linker histones control the gene expression by regulating the core histone post-translational modification. 2 of them we saw, many of them we have studied. But we have not published them. 2 of them we have published. So 1 is coming in Nature, it's in press. And these modifications are usually associated with heterochromatin. With this, I would like to acknowledge my PhD supervisor, Professor M.R.S. Rao, My postdoc supervisor, Professor Jeffrey Hayes from University of Rochester Medical Center, and my current supervisor, Professor Arthur Scalchi. He's very friendly. You can see my son shaking hand with him. He is at Albert Einstein College of Medicine, and he is the one who created all the knockouts for linker histones and showed that in mammalian system, H1 is essential for survival, otherwise death happens. So with this, I will thank you all for listening, and I will take any question.
So we are highly thankful for your insightful and valuable presentation, and I strongly believe that this will definitely help our students as well as other participants to further use your valuable comments in their research, and they can do well in the field of research, and especially in this area which you have elaborated so well, sir. So we are highly thankful for your valuable contribution to this international conference, sir.
Thank you very much. Thank you for the invitation. Do I leave now?